And wait, is everyone here like in 5th grade? I'm not exactly sure like the audience right now. Cause like if I don't know like which like if everyone knows what I'm about to explain. Can someone like just send me a chat? If not, I'll just move on. So here's some vocabulary to know beforehand in case. So there are oops. There are covalent bonds. So this happens when two molecules share electrons and they like connect with each other. So for example, water, because the oxygen shares oxygen shares the
We cannot see your screen.
what?
We cannot see your screen.
It says sharing. Wait, can everyone see my screen?
Like this.
Yeah. Okay. So this happens when there's a hydrogen, another hydrogen, and they are connected because they share electrons. Um, there are also there's also monomers. This is basically the building block of like larger molecules. In this case, a macromolecule. Um, so this is the building block of polymers. Of polymers. And in this case, this is going to be called an amino acid. And then there are polymers, as I mentioned just earlier. These are created by monomers, and um, so they're linked by covalent bonds, and in this case, they're called polypeptides. Um, and then there's going to be the macromolecule, which are just polymers created like folded together, and these are going to just be the proteins. Yeah. If there are like any questions, just let me know. What are proteins and why do we need protein? So protein, the word, comes from the Greek word proteos, which means first or primary, and that's because proteins are considered to be like the most important class of molecules, macromolecules. Um, and then there are other macromolecules like carbohydrates, lipids, nucleic acids. So, and then there's proteins. And most people know that proteins are needed for like muscle growth, and that's why you see people eating protein powder all the time. But they also function in like a whole and like other things like signaling, cell signaling, um, antibodies, and enzymes, and then nutrient transport, etc. So I'll write these down. So transport functions, transport like a channel protein. So let's say this is the membrane, and then you're allowing molecules to go through. There's also enzymes. So they often look like this. There's going to be like a substrate going in, and then they'll create a reaction. This will make a reaction go faster. I also mentioned antibodies, like defense. So like around the cell membrane, there's going to be protein sticking out like this, and they protect you against viruses. It's and etc. There's also structural, which is just muscle. Like that kind of. So that there's that. Okay. So protein structure, starting from the amino acid. So the amino acid looks like this. So it's gonna look carbon or the hydrogen and R group. And on its side, it's going to have a carboxyl group. And then on its left, it's going to have an amino group. So this is the amino group. And then this is the carboxyl. And then that's the hydrogen. And this R group. And then the R group will determine what it does, basically. It can be like a bunch of things. So there's a table that I have that I will, oh, I should have showed it earlier. That's okay. So, the amino acid can have like a bunch of different properties, like being non-polar. Which it is sharing. It does not look like this.
Yeah.
Like, can everyone see the amino acid drawing? I'm just, I don't know. Just let me know, like through chat. Can you even see chats? Okay, I guess it takes a long time to load. Sorry, let me get the chart real quick because it'll be difficult to see without it. So let me go back here. So this is the chart. Can everyone see the chart? Okay, well, yeah, this is the chart. And so there are nonpolar, there are positively charged, negatively charged, and they're polar. So nonpolar amino acids will form hydrophobic interactions, and then the partially charged and polar ones will create like hydrophilic interactions. And some amino acids also have disulfide bridges, which are when two sulfurs are bonded with each other. So here are the ones with sulfur. So there's that. Knowing this, it is now easier to visualize how these amino acids interact with each other to like fold into proteins. And there are four structures. So first there is primary. This is basically just a chain of the amino acids together. If it's easier to visualize like this. And then secondary. Also, they're connected by peptide bonds. And then there's secondary. This is when the amino acids like interact with each other and they create folds. And these are connected by the carboxyl and amino groups. So then they'll kind of look like this. So these are the alpha helixes. And then these are the beta pleated sheets. And then the next structure is tertiary. It's just more folding basically. This is when the R groups interact with each other, so I'll try drawing it out. So yeah, the R groups, okay, so this is looks like this kind of, and then there's going to be different interactions like, so one example, I'll just draw one. There's going to be hydrophobic interactions. Looks like this. Oh shoot, there's supposed to be. And then they're connected with each other. So this is hydrophobic because they share electrons evenly. Hydrophobic. There's also ionic bonds and also hydrogen bonds. And again, disulfide bridges. And the quaternary, that's going to be the last structure. It's not very necessary, but some proteins have it. It's basically just the tertiary structure, like multiple polypeptides just interact with each other to form like the final protein, which is like more complicated and it's just 3D. So quaternary. It's just more folding, basically. So the structure of a protein is extremely important because structure basically directly translate to function of the protein. And if any like part of your structure is wrong, then like it just won't work. Like imagine like trying to fit a wrong key into your like doorknob. It just won't work. And what can cause the structure to be wrong is the environment, the chemical, like its conditions, like the pH, like how acidic your environment is, salt concentration, how much salt is in your environment, concentration, the temperature, and more. So for temperature and pH, that's extremely important. Like for example, if I were to like fry an egg, like like an egg yolk, it's like protein, right? And if I like just straight up fried it, I can't just put it back together and like make it turn into a raw egg yolk again. Like that just won't work. But sometimes you can like uh like reverse denaturation. So this is called denaturation, like changing the shape of a protein. You can reverse it, but most times you can't. Um, there's also mutations. This happens when there's something wrong with the DNA and it like messes up the sequence of your nucleotides, and that will fold the protein wrong because the R groups will like interact with each other the incorrect way, and that will like cause protein shape to be different as well. And this will be explained last. Yeah, just mutations. So let's move on to protein synthesis. So protein synthesis happens in two steps. Oh, no, I don't want that. Transcription. And translation. So transcription, it involves DNA and RNA, and if you can recall that DNA looks kind of like this. There's like the base. So this is the phosphate group, the sugar, and then the nitrogenous base. So in eukaryotes, transcription and translation will happen. Transcription will happen in the nucleus, and in prokaryotes, it will happen directly in the cytoplasm. And so DNA directs RNA synthesis, and then it uses RNA to control protein synthesis, and this process is called gene expression. And so in the nucleus, DNA is going to it just looks like this in here. It's going to unravel partly because there's a protein called RNA polymerase that will attach to a region on the DNA, on like the gene, called the promoter, and it'll unravel slightly, like kind of like this. So now here are the bases. And then there's like the promoter on top. I'll draw that with like this. So RNA polymerase both unwinds the DNA and reads the DNA, which is like transcription. And it'll take like floating RNA molecules and start to create an mRNA strand, messenger RNA strand, from like here, like from one of the strands. Like it'll create this, and it'll grow outwards. Oh wait, I shouldn't have done that. Well, okay, it'll go that way. It'll decode from the template strand, which is this one, and then the template strand, and it'll create an mRNA strand. So let's say the DNA strand was ATGCGA. Let's say that's the DNA, and that's the 5 end. That's the 3 end. Then mRNA will be like 3U A C G C U 5 end. So once that's done, it's going to like modify itself like before it's finalized. And let's say they were like introns. Oh, my drawings are not showing up. That's weird. Are they showing up now? I guess I'll wait for it, or I'll reshare. Let me reshare. Oh, well, I'll just share again. Can everyone see it? Okay, so there will be some editing involved. Like let's say this strand was not needed. This is called an intron, and then that's just going to be removed. So now your strand is UACG5. Introns are important because it creates variety. Like it allows you to create different combinations so that your proteins are going to be like unique. Like you can create more stuff. When this is done, a methyl cap is going to be added to the 5' end. There's going to be like a methyl cap, and you're going to have your bases. And then at the end of the 3' end, you're going to add a poly A tail, which is just adenine over and over. And then G, C, A, U. So the methyl cap, the point of this is to initiate translation, while the point of the poly A tail, these are A's, is to help prevent like degradation of the RNA strand while it travels from the nucleus to the cytoplasm for translation. So now it'll go so now it's gonna start translation in a ribosome. Kind of like this, the ribosome. It's gonna be fed through here, and while it's being read, there are going to be tRNA, transfer RNA, bringing amino acids to be like connected to with each other to form the protein, to form the like polymer. So let's say it was G C A. It's going the tRNA is going to bring U G C, and UGC. So ACG codes for here's a chart. ACG codes for threonine. You can see here. Oh, by the way, these are called codons, and every 3 bases together is called a codon, and they'll code for certain amino acid. And this code is redundant because this allows for mistakes to happen, and it does happen sometimes. So this is like more of a fail safe. Also, everything will start with methionine, just like the start AUG, and then the stop codons are here, UAA and UAG. So yeah, this translates to threonine. Okay. So that's how the tRNA knows which amino acids to bring, because this is the coding strand basically, and then this is like the template strand, and then it's like this is called an anticodon, by the way. And it brings the amino acid that matches with this one. So then there's just going to be more and more different types of, like different tRNAs coming in and creating an amino acid strand until it gets to the primary structure, which is just like this now. And later it will fold, go into secondary, tertiary, and possibly quaternary to form the final protein. So what else? Also, DNA does not directly go to the ribosome. Like it would be theoretically easier, but because you cannot like just make DNA without like if every DNA went to the cytoplasm and possibly got degraded, you would just have like no more DNA. Like you have no more information left to make. So this is why RNA is very important. Now mutations. Oh, here. Just erase this. So there are point mutations and there are frameshift mutations. And within point mutations, there are 3 types of mutations called silent, missense, and nonsense. So silent, you can kind of guess what they are. Silent mutations, they end up this. So mutations are when there are changes in the DNA code, and that will obviously translate to having the wrong amino acid like sequence. So silent mutations are when the DNA code ends up coding for the same exact amino acid because, as I mentioned before, the DNA, the RNA, like for the to translate into an amino acid, it's redundant, and you can have like the same thing translated. So if you refer back to the chart, let's say I originally had TAT, but then a mistake happened, and then it ended up having C. There would be nothing wrong because it's codes for the same amino acid. So the next one is going to be missense. This happens when it codes for another amino acid. So let's say I had TAT again. Then I would somehow got TTT. This went from tyrosine to PHE. I'm not exactly know what it's called. And that's called missense. And then there's nonsense, where let's say I had TAT, it suddenly coded to a stop codon, which would be TAA. From tyrosine to a stop codon. Again, this changes the entire like structure of the protein because the amino acids will interact with each other in a different way. So then there are also frameshift mutations. And this happens when there are added amino acids, deleted amino acids, or just like shifted in a different way. So let's say originally I had, let's go back. Let's say my sequence was A, U, G, U, C, U, G, G, U. And a frameshift deletion happened, and G, so originally this translated to met, UCU, which was serine, and then glycine? I'm not sure. And let's say G was gone. Then it would be MET, SER, and then this was just JU. Like it wouldn't happen. Like it's just gone. So that's a deletion. Let's say there was an insertion instead. So like AU. So it would be AUA, which would make this UGU. Here the new one would be and then UGU, which is cysteine, and then CUG, which would be leucine, and then this would just not make sense. That would just not be coded, and that is like completely different from the original one. And then sometimes this like order will just like swap completely, which would again code for different amino acids, and those amino acids would interact with each other differently. I can talk more about protein functions if anyone wants, but that's about it. Uh, new drawings. If there's like anything that you want me to review, I can review that real quick because I know there's a lot of like delays. Okay, I'll just review all this really quickly. Um, okay, so starting from protein transcription, there will be an mRNA strand created. Let's say this is a 5N, 3N with like bases. So let's say this was based off of the template strand. So this would be essentially the mRNA version of the coding strand. What happened? Um, and then this will be will go through transcription, and then it will add a methyl cap and a poly A tail. This will go to the cytoplasm to find a ribosome. And then the ribosome is going to translate it basically, and then tell tRNA, which looks like weird clover things. I can't even draw it right. Let's just say it holds this, like an amino acid, and then it has anticodons. So let's say it was AUG, it would hold UAC. This would be met. It'll just keep on making these strings until like the primary structure, the polymer, will be formed. And from there, the polymers will fold into secondary, tertiary, and then quaternary if possible. Does anyone want me to talk about the bonds or not? I don't know. No reviews. And structure is really important. Again, reiterated. Then there's going to be mutations because that's what happens. It brings variety in our lives. Mutations, again, they change the structure. So by changing this code, which, well, not directly this code, it changes DNA code. So let's say this is DNA. Changes, so it's T, C, oh no, I'm tripping. A is C, U, C, etc. It changes this DNA strand so that like this could now accidentally be U, and this would be A, and then that would code for the wrong amino acid of this certain protein, and that would change the protein to be wrong. And then, yeah, that messes things up. So I'll talk about bonds. So starting off with covalent again, covalent is when two molecules share electrons because we love being stable, and you will want if you share electrons, you'll both become stable, and then that's why covalent bonds are formed. So then there are also ionic bonds. Ionic hydrophobic interactions. Disulfide bridges, hydrogen bonds, peptide bonds. What am I missing? I feel like I'm missing something. Let me double check. Okay, well, to start off, peptide bonds are in the primary structure. And then these are between the amino group, sorry, and the carboxyl group of each amino acid. And it looks like I'll try drawing it out to the best of my abilities. So let's say this is the R group. Hydrogen, carbon, double bond with oxygen. So this is one amino acid. And it's going to be connected to here. H, C, another R group. H, C. So this is the okay, let me get a different color. This is the carboxyl group. And then this is the amino group. So they're connected. Hydrogen bonds, you can see by the name, it's connected from hydrogen to another more electronegative group molecule. And I mentioned before that this works with oxygen and 2 hydrogen. And most of the time, oxygen is hogging the electron more. So yeah, hydrogen bonds. Disulfide, again, 2 sulfur together. I'm pretty sure they form a double bond. Very stable. And hydrophobic interactions. I already drew it, but it's kind of a bad drawing. And well, it's okay. Well, a property of it is that it doesn't like water, so there's that. And ionic bonds, that's basically when electrons are just taken away, kind of. And I can draw an ionic bond. Oh no, it's when opposite charges. Sorry, opposite charges. So this is an example of an ionic bond. This happens in the tertiary structure. So these happen in tertiary, this happens in primary. Oh, hydrogen bonds are also in secondary because they are formed between the amino and carboxyl groups. So yeah, there you have it. This is my first time giving a presentation of any kind, of any topic kind of. If there's need for any review, just let me know. Just text me if any questions. if no one has any questions, we'll end like now, I guess, and then you can like stay if you want to ask questions.